So hello students. So in the last lecture we have already completed up this conditions part, okay, which conditions are necessary for the germination. In today's lecture we are going to continue with the chapter, okay, of the seed structure. But in today's lecture we are going to perform of the experiment on the basis of water, temperature and oxygen. See, we have already studied about these conditions, but we have to prove whether these three factors are only necessary for the development of the seed into a plant. So now it's an experiment time. So our first experiment, as what was the first condition? The first condition was related to the water. So we are going to start up with the first experiment, which is related to the water only. We have to prove that the water is necessary for the germination process. So see students, we have to take a two beaker, that is beaker A and beaker B. Okay, inside this beaker B, just notice one thing. This is a cotton which is whitish in color, we can see out here. Okay, this whole is a cotton, but this cotton is quite dry. Okay, now again this is a cotton, and if you look it carefully, it is bit wet. Okay, the effect of this wet is bluish in nature, bit bluish, so that it can create up the difference for you. So the main difference is about the dry and the wet. In beaker A some seeds were kept, and in the beaker B also there was some seeds were kept. After some hours, in a day or two, not in hours I can say, in a day or two, we can see the seeds, okay, which were kept in the beaker A didn't germinate. Okay, didn't germinate. Okay, they are the non-germinated seed. We can't see any of the shoot part growing out. Okay, but as you can see in the beaker B, this was the seed in the start. After 2 days it started to germinate, and after few days it germinated with a big shoot system. Okay, we can see out here. So this says that the water is quite necessary for the seeds to get germinate. Okay, so who is going to absorb this water? Okay, who is going to absorb this water? Thus the water will be absorbed by the hilum, and I already completed this part. Okay, this is a seed, there is a whitish scar, and on the top there is a small hole, okay, which allows the water to come in, and because of this the the seed swells up, and when the seeds get swell up, so it starts to germinate. So the main difference would be dry and the wet part. Now the second condition was related to the suitable temperature. So we have completed this part. Okay, so what is the minimum, means the optimum amount of the temperature which is required for the seeds to get germinate? That is around 25 degrees Celsius to 35 degrees Celsius. Okay, this was the proper or optimum means a favorable condition in which the seeds can germinate fast. Now let's perform this exercise. This is a beaker A and this is a beaker B. Okay, inside this beaker A, first we are going to discuss the condition related to the beaker A. Inside this beaker A, we can see this white color ice cubes are kept, as we can see out here. These are the ice cubes which are kept inside this box. Okay, and the box is closed. Okay, now as there was some of the seeds we can see out here has been kept, as we can see some of the seeds have been kept. After a day or two we can see that seeds didn't germinate. Okay, so they have been proved to be a non-germinated seed. But in beaker B, if we keep, okay, See, there is a what cotton wool. Okay, here we have kept the seed out here. Okay, now this seed is kept in the air part. Okay, it is kept in the normal temperature. Okay, not too high, not too cold. It has been kept on the moderate condition. So we can see that the seeds have started to rupture after a day or two. Okay. Okay, I just wanted to give you a condition in the beaker B. See, the seed can germinate also. Okay, but the seed will show a sign of germination after many days. Okay, within a day or two it won't show any kind of a sign of germination. It will need around 7 to 8 days in beaker A to get germinate, but in beaker B it will start to show up the sign within a day or two. Okay, now the third question was related, the third condition was related to the oxygen. So we have to prove that the air is necessary for the germination. Now there are two flasks has been kept, that is flask A and flask B. In the flask A, first see, I'm going to mark this for you. In both of the flask, wet cotton wool has been kept. Okay, and the seeds have been kept, which has been perfectly soaked. Okay, the seeds are not dry, so the seeds are perfectly soaked up. Okay, now there is a test tube which has been inserted inside this flask. Okay, how it has been inserted? Actually it has been tied with a rope, and the rope has been kept out here, and this is a cork which has been fitted so that this test tube doesn't falls. Okay, this is something like this. Okay, it is not hanging in the air. So we can see there is a difference between those parts. In the beaker A, flask A, they have kept the plain water, but in the flask B there is an alkaline pyrogallic acid. Okay, see why they have kept the plain water and why they have kept the alkaline pyrogallic acid. I just wanted to show up a difference to you. See, water is perfect, both of them are kept in a suitable temperature, so two of the condition got favored. Okay, there are three condition in all, that is the water, temperature and oxygen. Okay, oxygen or air also you can call. See, two condition have been favored. See, this is wet, the water, the both of them are kept in the same temperature so that we can prove the exercise. Now the third condition, which is related to the oxygen. Inside the flask A, they have kept the plain water, and inside the flask B they have kept a solution which is known as the alkaline pyrogallic acid. After few days, in the flask A, the seeds will start to germinate. Okay, but in the flask B, the seeds won't show any sign, or a very slight amount of germination it is going to show. It is going to show a bit or no sign of germination. So why this happened with the flask B? Because inside this test tube, as we can see, there is a special solution which has been kept, that is the alkaline pyrogallic acid. The property of this alkaline pyrogallic acid is to absorb all the oxygen which is present inside this flask. See, if this is a flask, so definitely there would be a presence of the air inside this. Okay, suppose there would be the presence of air, but when they kept this alkaline, so the air which is present inside this flask has been absorbed, okay, by this acid. And because of this, the seeds got two of the condition, that is the water and the temperature, but the third condition that is of oxygen have not got these seeds have not got this oxygen. So that's why they showed up the sign of a there was a slight germination or no germination, you can call. Okay, so this proves that the oxygen means the air is also necessary for the germination part. But why they have kept this plain water? So that they have to prove whether this is the function, what is the function of this alkaline pyrogallic acid. Okay, see, this plain water has did nothing, but this alkaline pyrogallic has absorbed the oxygen. So we can prove now that this solution will absorb all the air which is present inside this flask. Okay, now a 3-bean seed experiment. This is quite very important. Okay, and this is interesting as well. See, this is a wooden plank. This is a wooden plank, as we can see out here, and inside this and there are three. They have took the 3-bean. Okay, the experiment is related to the 3-bean, so they have took the 3-bean. One of the seed is inside the water, one of the seed is in the air, completely in the air, and one of the seed is half in water and half in air. So see, this part of the plank is half in water and half in air. This is getting only water, this is getting only air. So we have studied about the condition that all the 3 factors are highly dependent on each other. Okay, we need proper amount of water, we need a proper amount of air, we need a proper amount of temperature. So what happens? See, first I'm going to discuss about this part, seed inside this water, this part. What happens when the seed is inside this water? See, and the temperature has been kept constant, so you don't have to think about this part. Okay, the temperature is quite suitable for all of the 3-bean experiment because they have been performed on the same day and on the same place. So temperature has been favorable for all of this. This seed, this first, has got the water and the temperature, but it does not got the proper amount of oxygen. So that is why the seed which is present inside the water didn't germinate. Okay, this was the first. What happens in the second? See, the temperature is favorable and it is getting a proper amount of the air, but this seed is not getting up the water because, as we can see, this wooden plank, this seed which has been kept is in the air. So it is getting out the 2 conditions, that is the temperature and the air, but it is not getting the 3rd condition, that is of the water. So that is why it is also not going to germinate. This seed, see, as I already mentioned, these 3 seeds have got the suitable temperature, so you don't have to ask up a doubt regarding that part. So the temperature is favorable. Okay, this half is inside this water and this half is above the water, means in the air. So this seed was getting a proper amount of all the 3 conditions, so that's why it starts to germinate. Okay, and this is a very famous experiment, 3-bean experiment. Now, as we have studied about the germination part, so we have to study about the 2 types of germination, that is the hypogyl germination and the epigyl germination. So what do you mean by hypo? The first one is the hypo. The word hypo means below, and the word gyl means earth. The word epi means above, and gyl, G-E-A-L, gyl is the earth. Okay, so first we will understand the terminology part. Hypogyl means below earth and epigyl means above earth. Let's see up this part. So this is a diagram which is going to help you out to understand this part in a more perfect way. The first condition is epigyl, What do you mean by epigill? Above the earth. To see, this was a cotyledon. This cotyledon started to, this seed started to germinate. This was a cotyledon, and this cotyledon is above the earth. We can see out here. Okay, this cotyledon is above the earth. See, where is the cotyledon? This cotyledon is above the earth. How we can say it is above the earth? Because it is the surface. Okay, and this cotyledon is above the surface. This cotyledon is above the surface. C means the cotyledon. So that is why it is called the epigill germination. Okay. But what happens in a hypogill solution? Hypogill germination. Hypo means below, gill means earth. See, we can see up this cotyledon. See, it started to germinate. Okay, and then this cotyledon, this is the surface, and this is the cotyledon. Okay, and this cotyledon is present below the surface of the earth. Okay, we can see this cotyledon is attached to the root system, this root system. So that is why it is called the hypogill. Hypo means below. Okay, below the earth, because it is below the surface. Okay, and this was above the surface here, and this was below the surface. So this was the two type of a germination. Now see, as we have completed this, as we have completed this epigill germination and hypogill germination, I just wanted to make you understand one of the terminology related to both of this part. See, where is the cotyledon? Here. So I am going to write C. And where is this shoot is starting from here, na? And where is this cotyledon? It is here, below the surface. And where is this shoot? Exactly above this part. So what we can see here, the C means the cotyledon is above the shoot system. Okay, here the cotyledon is above the shoot system. So that is why it has been called as epicotyle, because the word epi means above and cotyle means cotyledon. Epicotyle means, epi means the above and cotyledons, cotyle means the cotyledon. The cotyledon is above, right? Here the cotyledon is below, so that is why it has been called as the hypocotyle. Why hypocotyle? Because the word hypo means below, cotyle means the cotyledons. Okay, in the hypogill germination, the cotyledons remain in the ground. Okay, and the shoot systems elongates. Okay, the shoot system is elongated. In epigill germination, the cotyledon pushed above the ground. Okay, see, it has been pushed, so that is why this cotyledon has came up, and the region, okay, the shoot system is below this part. Okay, so here we can see the cotyledon is above and the shoot system is down, and here we can see the cotyledon is down and the shoot system is up. Now who show this epigill germination? This epigill germination is shown up by this bean seed. Who showed up this hypogill germination? This hypogill germination is showed up by the pea. Okay, these are the example. In exams they ask you, okay, who shows you the hypogill germination? That is the pea plant. And who shows up the epigill germination? That is the bean. Okay, even the maize shows up this hypogill germination. Now vivivorous germination. This is the third type of germination. We have already completed these two types of germination, that is the hypogyl and epigyl. Now we are going to learn about this viviporous germination. But this is a very special kind of a germination. This viviporous germination you won't be able to see in any of the it is not possible to see in all the plants. Okay, this type of germination is shown up by the special plants only. Okay, this special mode of seed germination is shown in the special plants only. The two examples I just wanted to give, that is the rhizopora. This is the rhizopora. This rhizopora is a mangrove plants. Okay, where does this mangrove plants we are going to see this mangrove plants we can see in the damp and the swampy places. Okay, damp and the swampy places means the land which is without the there is no land. Okay, only the water surface we are going to see in that part. This mangrove have been grown, and this kind of a germination is shown in the rhizopora and soneraceae. Okay, now first we will discuss what do you mean by the word viviporous. The word viviporous is a special type of a term. Okay, here we can see this we can see in the tomato also. This is an example of a tomato also, but according to your syllabus you should write the soneraceae and rhizopora. This is the only explanation part, so that's why I am giving you as a tomato, but you do in the exams you don't have to write tomato. See, inside this tomato we can see okay, inside the tomato there are many seeds. Okay, so what happens when these seeds get the favorable condition inside this, so they start to germinate. We can see, so the word viviporous germination means the seeds which germinate inside the fruit while it is still attached to the parent plant. See what we usually do, this is a tomato suppose, and these are the seeds, and if we provide them these seeds get the proper amount of conditions, so they can start to germinate, right? This is the basic condition. But inside this, suppose this is a tomato, and inside this the seeds. Okay, when the seeds which are present inside the tomato get of the favorable amount of the conditions, they starts to germinate. Okay, and they are still attached to their parent plant. Okay, they have not been detached. Okay, they are inside this only. So this is called as the viviporous germination. The word viviporous you have already heard in your lower standards. Viviporous means producing live young ones. Okay, that means giving birth to the young ones. There is an oviporous also. What do you mean by oviporous? Which gives egg, like how the hen gives the egg, and inside this egg there is a little chick. Okay, but viviporous means giving a birth to the young one, as in the condition of a humans. We don't give egg, we give a birth to directly the young one. So that is why this kind of this special name has been given to this part, that is the viviporous germination, because it gives the germination condition inside the plant only. Now we are going to learn about the seedling condition. So see, before starting of the seedling, let's understand this diagram. See what we are going to do. We are going to insert a seed first, and we are going to provide them the favorable condition. And when we provide them the favorable condition, it will start to germinate. It will convert into a sprout. But when this sprout means a condition which is not fully developed. But when this seed gets converted into a seedling, so it develops into a new plant. We can see, suppose this is a soil surface, we can see a proper amount of a root system. This is a shoot system, and we can see the leaves also out here. Okay, we can see the proper amount of the leaves also. So let's understand what do you mean by seedling condition then. Seedling is a stage in the growth of a plant from a seed. Okay, so as I have already said, Okay, there was one seed. It started to germinate, and when it germinated fully, it got developed into a new plant. No? So this is nothing but the condition of the seedling. Now from a seed, before it has become wholly independent of the food stored in it. What does this line means? Now see, this seed has developed into a plant. Now this got the proper amount of leaves. Now it does not have to take the help of the endosperm to take up the nourishment. Now this plant can manufacture up its own food with the help of the leaves, because we know this photosynthesis, where this photosynthesis occurs in the leaf part. Okay, this leaf takes up the sunlight, and then the photoreaction happens inside this leaf, and this is how they can make up their own food. When the seedling get developed more, okay, and becomes a mature plant, it start to produce a flower, and that flower will turn the ovule into a seed, and in its turn means the procedure is going to happen. Okay, this is a seed condition, germinated seedling condition. Okay, this is a seedling condition, and now from this a mature plant will take place, and the mature plant will give rise to a flower. The flower will give rise to the seeds, and this is how the hereditary function, okay, the hereditary function or the next generation takes place. So this was all about the seed structure. Thank you, student. This was the part 2.